The purpose of the standard operating procedure is to describe the procedures for collection of representative soil samples. Analysis of soil samples may determine whether concentrations of specific pollutants exceed established action levels or if the concentration of pollutants present a risk to public health, welfare, or the environment. Soil samples may be collected using a variety of methods and equipment depending on the depth of the desired sample, the type of sample required, and the soil type. This video will display an example of soil sampling using the T-handle auger method. The equipment used for this soil sampling method consisted of the following: a plot plan, string, a random number generator, tape, survey stakes, a camera, appropriate sized glass sample containers for sample collection, Ziploc plastic bags, paper towels, logbooks, labels, chain of custody records and custody seals, field data sheets, sample labels, ice cooler, an ice pack, deionized water, a trowel, and a T-handle auger. Safety equipment included latex gloves, safety vest, closed-toed shoes, and long pants. Reagents were not used for the preservation of soil samples in this example.
To perform a standard operating procedure, first perform a general site survey prior to site entry. Specific site factors need to be included when determining the sample location, including the extent and nature of the contaminants. Once the site is established, stakes may be used to identify the sampling location. In this example, a 10-foot by 10-foot grid west of the Peterson building was plotted on the Texas A&M University campus. To begin sampling, decontaminate equipment using deionized water and ensure that it is in working order.
Samples are collected from the near-surface soil using a trowel. Surface material is removed to the required depth, and a T-handle is then used to collect the sample. First, attach the auger bit to a drill rod extension and attach the T-handle to the drill rod. Clear the area to be sampled of any surface debris, such as twigs, rocks, and litter. It may be advisable to remove the first 3 to 6 inches of surface soil for an area approximately 6 inches in radius around the drilling location. After reaching the desired depth with the auger, slowly and carefully remove the auger from the hole. When sampling directly from the auger, collect the sample after the auger is removed from the hole and proceed. Place the sample into a stainless steel, plastic, or other appropriate homogenization container and mix thoroughly to obtain a homogeneous sample representative of the entire sampling interval. Then either place the sample into appropriate labeled containers and secure the caps tightly, or if composite samples are to be collected, place a sample from another sampling interval into the homogenization container and mix thoroughly. When compositing is complete, place the sample into appropriate labeled containers and secure the caps tightly. Decontaminate the auger and tube sampler between samples. Abandon the hole according to applicable state regulations. Generally, shallow holes can simply be backfilled with removed soil material.
There are no specific quality assurance activities which apply to the implementation of these procedures. However, the following quality assurance procedures apply. All data must be documented on field data sheets or within site logbooks. All instrumentation must be operated in accordance with operating instructions as supplied by the manufacturer, unless otherwise specified in the work plan. Equipment checkout and calibration activities must occur prior to sampling and operation, and they must be documented. When working with potentially hazardous materials, follow U.S. EPA, OSHA, and corporate health and safety procedures in addition to the procedures specified in the site-specific health and safety plan. Chemical preservation of solids is not generally recommended and was not used in this demonstration. Samples should, however, be cooled and protected from sunlight to minimize any potential reaction. Store the samples in an ice cooler with an ice pack.
There are 2 primary potential problems associated with soil sampling: cross-contamination of samples and improper sample collection. Cross-contamination problems can be eliminated or minimized through the use of dedicated sampling equipment. If this is not possible or practical, then decontamination of sampling equipment is necessary. Improper sample collection can involve using contaminated equipment, improper labeling, disturbance of the matrix resulting in compaction of the sample, or inadequate homogenization of the samples where required, resulting in variable, non-representative results.